So this is the seagrass biology session. I'll be chairing it and kicking us off as well. Yeah, a little bit of a miscellaneous session, a bit of a mixed match of talks, but it should be really fun. For those of you that don't know me, I'm Matthew Fraser. I work at the University of Western Australia. I'm a research fellow there, and I've been a seagrass addict for going on 10 years now. And I work on primarily microbial ecology and seagrass ecosystems. So to kick off the seagrass biology session, we're talking about microbiology. Good start. Yeah, so in terms of microbiology and seagrass ecosystems, it's been a little bit of a black box for a while now. Until recently, we've known that inside seagrass meadows and the sediments, there tends to be a higher bacterial diversity than external bulk sediments in unvegetated areas. And we know there's novel diazotrophs, really important for nitrogen fixation. And in general, if we have intact microbial populations on our transplants, they tend to survive a little bit better than those where we disturb the microbial communities. But apart from that, a little bit of a black box. But we do know that microbes play a really critical role in biogeochemistry and the health of macroorganisms in general, based on terrestrial environments, coral reef environments, and a whole range of studies. And even Glenn Hines' talk yesterday, using nanosims with Flavia's work, really showed that in a beautiful way, the uptake of nitrogen, the breakdown, the mineralization of it by the bacterial communities on the leaf surface, and then the entry of that into the seagrass tissue itself. So really cool stuff. And as Peter was mentioning earlier, the sequencing revolution is really changing the way we look at microbiology in seagrass ecosystems. So the cost of molecular sequencing is getting a lot cheaper, which is great for us because seagrass ecologists don't always tend to be cash rich. So my particular way of looking at microbial communities uses a technique called shotgun metagenomics. So Peter, again, really topically mentioned this morning. And basically with metagenomics, we look at the DNA profiles of the entire microbial community in our environmental sample, which allows us to see the entirety of the genes that our community possesses. So we go out, we collect our sample, whether it's in the water column, leaf biofilm, sediment, whatever it is, we collect the sample, extract and purify the DNA of the microbial community, and sequence that in a machine like Illumina HiSeq. And what's really cool about metagenomics is that it provides a taxonomic and functional characterization of the microbial community. So we don't just get to see who's there, but we get to see what they do as well. And it also isn't targeted, so we get some quantitative data on the rare taxa and the rare functions that our microbial community can carry out. So where I come in is trying to use this tool of metagenomics to assess how seagrass-associated microbial communities change across environmental gradients. And I'm going to look at two different case studies based in Western Australia, one beneficial, one negative impacts that microbes are having on seagrasses. So the first example is from the Shark Bay World Heritage Area in Western Australia. And what's really cool about Shark Bay is that it's got a really strong permanent salinity gradient that ranges from 35 parts per thousand in the north to over 70 parts per thousand in the south. And the seagrasses occur up to around the mid-50s. So we sampled the microbial communities associated with the seagrasses across this salinity gradient. Now, at the same time as we've got the salinity gradient, we've also got a gradient in the phosphate concentrations in the water column. And Shark Bay is ultra-oligotrophic. So by the time you get down to the hypersaline areas, you can't measure phosphate in the water column using traditional, conventional methods. So really, really extreme place to grow if you're a seagrass, but they form these productive, beautiful meadows, carbon hotspot, as we've heard already. We also looked at simple biogeochemistry, enzyme analysis across the same sites, and working in amphibolous Antarctica meadows, so long-lived, relatively long-lived temperate species. I'm going to jump straight into the data, time poor. So this is a simple PCA plot. The closer the points are together, the more similar the taxonomic profiles are in those communities. And it's color-coded based on salinity, with the blues being the higher salinities, the green being the low salinity. And you can kind of see that there is a clustering based on salinity. And if we look a little bit more detail, we can see certain taxa that change between our high and low salinity sites. So our proteobacteria tend to be underrepresented at the high salinity sites and become replaced by planktomycetes. They're significantly more abundant in the hypersaline, low phosphorus areas. And what's cool about this is we know based on macroalgal literature that this particular microbial group is really important for nutrient cycling on the biofilms of kelps. So we're beginning to see the kind of roles that they might be playing. But as I mentioned earlier, metagenomics lets us look at that in a lot more detail. So this is us looking at the functional community instead of the taxonomic profiles that we looked at earlier. Also clustered by salinity. What we've seen in Shark Bay was that genes related to photosynthesis and DNA metabolism were significantly more abundant at our high salinity sites. Nitrogen metabolism was higher at our low salinity sites, but what was really cool for this story was that phosphorus metabolism genes were more abundant at the low phosphorus, hypersaline sites. So the increase along this phosphorus gradient. And just like Peter was talking about earlier, we can actually begin to look at the individual genes that are changing in our communities instead of what's happening inside the seagrasses. This is the microbes. And the ones I've highlighted here are the phosphorus metabolism genes that increase in abundance along the salinity gradient. And these all had in common the fact that they were related to the metabolism of organic phosphorus compounds. Now, based on other research in Shark Bay, we know that despite phosphorus becoming lower in the water column, organic phosphorus accumulates in the sediments at those hypersaline areas. And these microbial communities are targeted to access that phosphorus pool. However, we can't always infer the expression of genes based on their abundance. This is just DNA, so it's showing what the community could do, but it's not transcriptomics. It's not actually showing what they're doing. This is normally where you'd go and do metatranscriptomics. I didn't have enough money. So instead, this is where the enzyme expression come into things. And here, I'm just going to show one enzyme that we looked at, alkaline phosphatase, which is really common in marine sediments. It's involved in the breakdown of organic molecules, phosphate monoesters, essentially releasing phosphate, which is then available more readily for biological uptake. And what we can see is, as we increase along the salinity gradient, we get a much higher expression of alkaline phosphatase from the sediments. And this was strongly correlated with our gene abundances for this particular enzyme, which led us to believe that phosphatase, at least these phosphatases, were most likely produced from microbial sources. Now, seagrasses can also excrete alkaline phosphatase, but it's pretty energetically costly. And if you've got microorganisms living around you that can do the job for you, that's a win-win situation, especially when you're releasing organic exudates yourself. So what we think is happening in Shark Bay is we've got our low salinity, relatively high phosphate in the water column, and the microbial community doesn't really need to do much up there because there's enough phosphate in the general environment. Whereas when we go down the salinity gradient and phosphate in the water column becomes really, really low in concentration, the ecological role of this microbial community in breaking down sedimentary organic phosphorus becomes much more important and may actually supplement the productivity that we see from the seagrasses in these areas. So that's a potential benefit that microbes produce for seagrasses. What about the other side of the story? So here we're going a bit further south. This is Coburn Sound, just off of Perth metropolitan area. Really famous place in terms of seagrass ecology because there's a large decline of seagrasses there due to coastal development between the 60s and the 80s. Things have improved since then, but we're still seeing localized declines in certain areas, and we think that's now driven by sulfide intrusion. So here we wanted to look at, we had previous data on sulfide intrusion with stable isotopes, and we wanted to look at how the microorganisms were also changing across this gradient of the seagrasses being impacted by sulfide intrusion from sediments. And that sulfide intrusion is correlated to long-term shoot declines over about 10, 15 years based on long-term monitoring data. So again, we've done similar metagenomic analysis as the Shark Bay example I showed you, but this time we paired it with our sediment biogeochemistry measures and our seagrass measures as well. So primarily looking at delta 34S, the stable isotope of sulfur in seagrass tissues, and F-sulfide, which is a measurement effectively of how much sulfide is entering the plants from the sediment. And we know sedimentary sulfides, if they enter seagrasses, they can be toxic. And this is Posthonia sinuosa meadows this time. Again, long-lived temperate species. Another PCA plot. Again, we've seen clustering of microbial communities based on what sites they're from. And we've generally seen that those impacted previously with sulfide intrusion in the red circle, they tended to cluster together, while those that weren't impacted were a little bit different, with one sample doing something else. And this graph hasn't turned out really well on the screen, but basically what we're doing here is looking at our sulfate reduction genes in particular. So this is going back to the functional side of things, and comparing that to leaf F sulfide, which this is the proportion of sulfides that enters the plant from the sediments. So higher value here, more stressed seagrasses. And what we can see is, in general, we've got a positive relationship. As you increase the sulfide stress in the seagrasses, you also tend to get a higher sulfate reduction percentage in your microbial populations in those sediments. Now with metagenomics, we can go back and look at the individual taxa that could be playing an ecologically important role here. So these are two of our sites that were highly impacted, Warmby Sound and Garden Island. And we can begin to look at the certain taxa. So I'm not even going to try and pronounce them, but basically these guys are purple sulfur bacteria, and we've got the sulfur bacteriaceae there. They're sulfate reducers. So again, these guys we know have an active role in sulfur cycling, and they are correlated to our sites of sulfide intrusion. So what we can say is that microbial communities are responding to the same sulfur gradients that our seagrasses are. We're not quite at a point where we've got cause and effect yet. We really need to start doing some manipulative experiments in labs before we're at that point, which is quite difficult with microbial communities. But we're seeing some really interesting patterns out in the field. And this leads us to, pie in the sky sort of ideas. Can we use sediment microbial communities as potential indicators of sediment health? Sediment health is hard to measure and incorporate into management plans. So maybe this gives us an avenue to do that. And potentially one day, could we condition sediments to increase survival of transplants or of seeds for restoration potential? Is there particular families or taxa that we can inoculate sediments or organisms with to increase the likelihood of survival? So to wrap up, the rhizosphere microbial communities in seagrass meadows, they do respond to environmental gradients, and this in turn, through their role in biogeochemistry, can change the growing conditions for the seagrasses themselves. Our next steps, what I'd really like to do is link this microbial diversity to actual indicators of seagrass health. That feeds really nicely into what Peter was talking about earlier, things like the transcriptomic approach, proteomics, that side of things becomes really cool. Again, as I mentioned earlier, using the microbiome to inform seagrass management and restoration might sound far-fetched, but they do this already in terrestrial environments. So why can't we do it in seagrass meadows? And finally, do we need to consider the seagrass holobiont? So not just the seagrass itself, but its associated microbial communities, where we're considering how seagrass resilience might change into the future with increasing temperatures, changing light availabilities, or freshwater inputs. That's me. I'll finish talking now. Thanks very much. Since we've got no talk after me, I think we've got time for questions. Gary? Two seconds, Gary.
So you've shown relationships with phosphorus availability.
Yep.
And responses, potential responses, because again, potential responses in both enzymes and responses to enzymes, potential responses in the micros and macros.
Mm-hmm.
The one thing that's missing there is actually something that would link the production of phosphate or inorganic phosphorus to production in the seagrass. If you were sort of going and doing a sort of an experiment where you actually add labeled phosphorus.
That would definitely be the next step. So try to stable isotope approach, not quite using radioisotopes, which are a lot more difficult, especially when you're working in a World Heritage Site, to release those into the environment. We tried to use, so with phosphorus, it's a lot harder to track the nitrogen using stable isotopes, but you can look at, if it's phosphate you're interested in, you can actually look at the oxygen that's on those phosphate molecules and track it with 18O instead of looking at the phosphorus itself. We tried that. That method is used extensively in terrestrial ecosystems. We couldn't develop a methodology that got it to work in seagrass ecosystems that would have beautifully shown how much of the seagrass phosphate that it takes up is actually linked to the production by the microbial communities across that gradient. We think we're not entirely sure, we think it's because of the low concentrations of phosphorus in shark base. It's a hard place to develop the methodology. Salinities as well, and also some of the compounds that seagrasses naturally have can interfere with some of the, there's a big extraction process you have to go through. Sorry, Ella. Big extraction process that you go through to purify and isolate that oxygen from the phosphate. So we think something in the seagrass is interfering with that. It's definitely a cool area for research, but yeah, I agree with your point. Any other questions?
What is the primer you use to unravel the metagenome, microbial metagenome? What are the primer you have used for this?
So with the metagenome sequencing, you just extract the DNA.
What are the target primer you used for unravel the metagenome?
So you don't effectively need a primer itself, you just extract the DNA and...
You have shown the microbial community there.
So it's bacteria, archaea, fungi.
There is no mention of archaea anywhere in your presentation.
Yeah. I have
only proteobacteria and planktomycetes are the dominant cluster you mentioned.
Yeah.
But as he asked, you work only for salinity gradients. What about the biological productivity of that particular region?
Sorry, what was that?
Biological productivity.
Yeah. So in terms of the seagrass productivity or the micro?
Biological productivity, especially in sense of primary producers in that particular region. Then only, because why I am telling this is, when a ecosystem is represented by high factors of proteobacteria, it indirectly indicates that that region is very particularly productive ecosystem biologically.
No, I wouldn't say that.
It is there. Proved evidence are there for that. Even that we done the same work in Antarctic waters, and we find that if you take in the Pacific Ocean also there is work, in the Red Sea there is work. You just mentioned this, especially of the diversity of proteobacteria. You can equate to biological productivity of that particular ecosystem.
Yeah, look, you can definitely go into detail about the individual groups with metagenomics. I have done, it's just it was limited time to talk.
It's a very interesting piece of work.
Yeah, I can definitely talk to you about that later.
I appreciate your work.
Yeah, in terms of the productivity question, I think it's the environmental controls in the microbial community that also have an impact, not just productivity per se. Hi, Peter.
Quick question.
Yes.
I like the application, it was great. I'm struggling in my mind, and I'm not sure if Casper's here, he may be able to help me out. The rhizosphere. So you're going to have oxygen coming out of the roots.
Yes.
You've got plenty of sulfur in the sediment. You've got increased sulfur-reducing bacteria. Why the hell are they in that space when the roots are protecting themselves? Your samples were in the rhizosphere, not inside the roots, where you got the increased bacteria?
Yeah, so I think it's a spatial scale. I think if you sampled the endophytic community, it would be very, very different from the rhizosphere community, which would be very, very different from a centimetre to 10 centimetres away.
Okay.
So I think it's very much a spatial scale problem. This is a very broad approach using metagenomics. I think you could definitely use these techniques, in lab systems and across much more controlled spatial scales and get some really interesting stuff. To me, this is almost like exploratory analysis that you can take further with more targeted approaches.
Good. No, it makes sense now. I was just struggling.